What's up, hooligans, and welcome to another video. In this video we're going to talk about DNA translation.
DNA consists of two polynucleotide chains that wrap around each other in a double helix. Each nucleotide has a nitrogenous base. Cytosine and guanine bond to each other, adenine and thymine bond to each other. Since each nitrogenous base can only bond with one counterpart, it allows for DNA to temporarily separate while an enzyme unzips the DNA to allow for transcription into mRNA.
Translation takes part in the ribosomes, and it is the last step in gene expression. It is crucial for cell division and cell proliferation. In the process of translation, a cell reads information from a molecule called messenger RNA, and it then uses this information to build polypeptides, also known as proteins. What is mRNA? Well, one could say that mRNA are instructions for building polypeptides. mRNA are in turn made up of 3 nucleotides called codons. Here are some key features of codons to keep in mind as we move forward. There are 61 different codons for amino acids. 3 stop codons mark the polypeptides as finished. One codon, AUG, is a start signal to kick off translation. It also specifies the amino acids methionine. mRNA is an exact copy of one strand of DNA, but thymine is swapped out for uracil.
The process of translation has 3 parts: initiation, elongation, and termination. First is initiation. For anything to happen, you need a few things: a ribosome, a mRNA, as well as a tRNA carrying the first amino acids in the protein. Together, these parts create an initiation complex, a molecular setup needed to start the translation process. The ribosome assembles around the target mRNA. The first tRNA is attached to the start codon. The second part is elongation. This is the stage where the polypeptide chain gets longer. The tRNA transfers an amino acid to the tRNA corresponding to the next codon. The ribosome then moves, translocates, to the next mRNA codon to continue the process, creating an amino acid chain. Lastly, there's the termination. Termination is when a codon that does not translate into an amino acid enters the translation site. The chain of amino acids, the polypeptide, ends. And now we have a fully formed protein. And now, lastly, a summary of what we have gone through.
Translation is the final step of translating DNA into functional proteins. Ribosomes attach to the modified mRNA strand and translate that strand into a chain of proteins. This is accomplished by tRNA that carries specific amino acids to the ribosomes, where 3-letter codes are read and matched to their specific amino acid. Once the chain is synthesized, it usually folds into conformation that is functional. Single DNA mutation can be disastrous, but the DNA mutation is transcribed into a 3-letter mRNA code that codes for the wrong amino acid. This prevents the amino acid from forming into a functional protein.